Let's take a look at some classes of chemicals that are important for immune responses. The first one, acute phase proteins, you will not hear much about in this lecture. They are there to enhance inflammatory responses. Chemotaxins, those are molecules that attract phagocytes and direct them to a site of infection. Cytokines, those are mostly growth factors and also signaling molecules that affect the activity of another cell. Opsonins, those are coating proteins. They will coat a pathogen and it makes it that much easier for a phagocytic cell to eat those bacteria then up. So they're basically labeled bacteria, but they have opsonins on them. Pyrogens are fever-producing substances. And here is a long laundry list of other terms and chemicals that you should know about. So let's go through them and I'll tell you which ones are important. Antibodies, of course, immunoglobulins, you definitely should know about antibodies. We'll talk more about them. Bradykinin is a strong vasodilator that we sort of marginally talk about. Complement is a system that we'll be talking about pretty much next. C-reactive proteins we don't need, granzymes no. Heparin is an anticoagulant. That was part of the blood lecture too. Histamine, another vasodilator, is responsible for allergic reactions. It's what these mast cells and basophils are filled up with. Interferons, we just talked about them. They help with defense against viral infections. Interleukins, that's a very general term of a group of molecules that are used as communication molecules between white blood cells, therefore called interleukins, between white blood cells. Kinins, such as bradykinin, so that's what you had up here. The kinins in general, they are vasodilators typically, and so we're not going to talk much about those. Lysozyme was this enzyme in your mouth that weakens the cell walls of bacteria. So that you should know. Major histocompatibility complex, the MHCs, we're going to talk some more about them. That helps with self-recognition as opposed to foreign recognition. The membrane attack complex, it's part of the complement system, and you will see that in a minute. Perforin, that's a protein that makes membranes leaky and helps basically poke holes, therefore the name perforin, poking holes into cells. A superoxide anion, that is a really powerful oxidant and helps to destroy bacteria or cells that you don't want. Now the T-cell receptors, that is very important because T-cell receptors, they help with the communication. So if you have some interleukin, it needs to bind to a receptor and then communicate what this T-cell, for example, needs to do. It's important for T-cell activation. And then tumor necrosis factor, those are cytokines that promote inflammation and then induce apoptosis. So what we need here would be the T-cell receptors. We will talk about them. And sort of the tumor necrosis factor, well, we don't really talk about that one that much more. Next is the complement system. That is basically a set of enzymatic reactions that sequentially activate one player after the other until you have the membrane attack complex. So basically it resembles to some extent the clotting cascade, where you have sequential activation of different steps, and once everything is in place, then you're going to have the wanted result here. Now, the main three results of the complement system would be opsonization. That means labeling of a pathogen so that macrophages have an easier time eating it up. Enhancement of inflammation is another result of the complement system. And then very important is cytolysis. That means we're going to drill a hole into the surface of the bacteria, and then the cell contents will leak out, and then you're lysing the cell, basically. The easiest way to activate complement system would be if you have already an antibody that has found its target. So here's your bacterium, your pathogen, and we already have an antibody, this Y-shaped structure right here, that has bound to this microbe. Now, the first component of complement, that's called C1, that one will bind to the stem region right here of an antibody that has found its target. So as soon as the arms of this antibody here binding to their target, they're exposing a binding site down here. So the molecule, the antibody will change its shape on the stem structure and then expose a binding site for the first component of complement C1. Once that has occurred, then we will cause C2 and C4 to split into their subcomponents, C2b and C2a, and C4 will split into C4b and C4a. Once that has happened, you're going to activate C3, which is the third component of complement, and once that has happened, there's no more holding back. Then you will get the result of opsonization and inflammation enhancement and cytolysis. So here, basically the steps continued. And here, so once we have the all-important C3 activated, we will get the opsonization, we will get the enhancement of inflammation, which then draws more white blood cells to the area, and we will get the membrane attack complex, the cytolysis. So, well, basically here, this is pretty interesting actually. We're going to assemble C5 through C9, these components of complement, and it will drill a hole into the microbe, and then all the contents are leaking out, as you can see here. So basically, once C3 has been activated, it will split into C3a and C3b, and that then activates C5 through C9 to form this membrane attack complex, and then we're activating mast cells to release histamine from these granules inside of the mast cell, and we're going to enhance phagocytosis by coating bacteria with C3b, which is this component right here. Here is the membrane attack complex, and nice and big. I like this picture. It's pretty cool. Think about it. The C5b through C9 will be assembled and form this ring, and it's drilling a hole into the microbial surface, and then you're getting basically the contents are leaking out of this bacterium, and that is known as cytolysis, and it will kill the cell. So here, another representation of the same thing. Here you have your pathogen right there, and you have this membrane attack complex that's forming these pores, these large holes in the surface of the microbe. So the membrane attack complex has formed, and then here you can see how this bacterium has all sorts of holes there. Contents will leak out, the cell will swell and lysis, and it is dead. Now let's take a look at lymphocytes. They can differentiate into B lymphocytes and T lymphocytes. B lymphocytes, they make antibodies. If they're actively secreting antibodies, we call them plasma cells. If they're sort of dormant and just sticking around after having produced antibodies of a specific kind, then we call them memory cells. But so B lymphocytes, they're associated with antibody-mediated immunity. They make antibodies either currently, or they know how to make the antibodies, so they're either plasma cells or they are memory cells. Activated T lymphocytes, they develop into either cytotoxic T cells that then attack and destroy virus-infected cells, or helper T cells that regulate other immune system cells. And we have another type of lymphocyte, the natural killer cells. We talked about them earlier. They attack and destroy virus-infected cells and also tumor cells, cancer cells. Now, lymphocytes, they're all part of your third line of defense. So these are antigen-specific responses. This is definitely acquired immunity, very specific immune responses. This type of acquired immunity can be either acquired actively or passively. Actively means that the body is exposed to the pathogen and produces its own antibodies. Now, you can do this either by just getting sick and going through the process of making antibodies, and then acquiring that kind of immunity because you know how to make the antibodies, or you can get vaccinated, and that sort of it's a shortcut, and you don't have to get sick, and you still have the antibodies in case your body is faced with that same antigen again. Passive immunity means that you acquire the antibodies from another organism. That is something that they're currently talking about with the coronavirus, that they're using the plasma, which contains the antibodies of those people that have recovered from coronavirus. And so now they're using those antibodies to help another person recover. So those would be that will be passive immunity. You're given the antibodies by another source. It also happens when you're nursing a baby. The mother's milk contains incomplete antibodies, but still it helps. Okay, so here let's take a look at primary immune responses, how the exposure to an antigen triggers the formation ultimately of antibodies. So here is your antigen, and it's the surface of a bacterium right here, and you will activate B cells to differentiate, and then they, by trial and error, they need to make an antibody that fits the red circles right here. So that's called clonal expansion and clonal selection. It's a pretty elaborate procedure that involves splicing of the gene that makes the antibodies, and it's pretty complex, so we're going to take a shortcut to all of this. The end of it is that you're going to have plasma cells that can make the antibody, and once you don't need the antibody anymore, then you're just going to keep a memory cell around, and these memory cells, they are very long-lived. Preferably you can keep them for a lifetime, so that, for example, childhood vaccinations that you received, many of them last you a lifetime, and you will be protected because you keep these memory cells, and once you see that same pathogen again, then you're ready. You can convert the memory cells back into plasma cells, and you will make your antibodies that you need. So here, this would be B lymphocytes. They're secreting antibodies, and again, a B lymphocyte that actively secretes antibodies is called a plasma cell. Once you don't need these antibodies anymore, here's churning out these antibodies by the thousands per second. Once you don't need these antibodies anymore because you got well, then we will turn these plasma cells into memory cells. So plasma cells can be easily converted into memory cells, and memory cells can be reconverted back into plasma cells very quickly. So here would be a primary immune response. You're exposed to the antigen either naturally and then you're getting sick, or you got vaccinated right here, and so now it will take you about a week before you have plasma cells that are churning out the correct antibody. And so then the antibody concentration is going up right here, and if you were exposed to the pathogen, you were sick here, and then you're starting to get better right here. Now, if you have a fast-killing pathogen, like Ebola virus, for example, this is not enough time, then the virus wins, and you may not survive this. But hopefully your immune system is making the antibodies quickly, and then you don't have to you will get better. And if it was a vaccination, then you don't have to get sick at all, and you're just making the antibodies and you're ready. So if you have a secondary immune response, then that means we have these memory cells. So memories are already there because we had a previous exposure to this pathogen. Now, all it takes is we're going to convert the memory cells back into plasma cells. The plasma cells are churning out the antibodies really fast because they didn't have to go through the clonal selection process. There was already a memory cell that knew how to do the right antibody for this pathogen. And now, right away, the second exposure to the same pathogen, the antibodies will be present almost immediately, and your secondary immune response is it's really fast, and see the concentration of antibodies is really high. Now, here a word about, in case if you are an anti-vaxxer or somebody that knows somebody that's an anti-vaxxer, think about it. Vaccination is awesome. You will basically, it's like getting the final exam 2 weeks in advance. You are ready. You're prepared. And then whatever pathogen here comes along, your second exposure, you have the memory cells that you can convert into plasma cells, and now you're ready. So vaccination is really great, and it gives your body the chance to have exposure to a pathogen, a dangerous pathogen typically, that without having to get sick, you have the time to make the correct antibody. Then you put that antibody or these plasma cells, you convert them into memory cells, and if you see that pathogen again, now the real life version, and you're ready. you have the antibodies available almost immediately and you don't have to get sick at all.